and I must say that first lecture was quite fulfilling for me as well as a expert speaker, because you guys asked so many questions. I was happy. Until now I am receiving lots of questions, which shows that I could connect with you. So the second lecture is slightly different. So the first talk was a bit more generic on what we are learning in the what are the plus and minus, where we encounter challenges, how we overcome those challenges, and this is on a real data, and I will again narrate different kinds of challenges that we encountered here. So the purpose is twofold for this talk. You can see that it is not traditional medical imaging sort of an application, but we are now talking about the genomics data. And as an AI ML or a data scientist, how you can analyze this sort of data is an interesting part that you should be excited about seeing it. The second is, because it is on the real data, so you will see that there are practical challenges when we are handling the real data. I think the audience is still not settled, so I'll wait until everyone settles. Okay, so I'll begin. So this is the team. Vivek was my PhD student and is now doing postdoc at Columbia University, USA. Professor Ritu is my collaborator at the hospital, All India Institute of Medical Sciences. She is also heading the lab oncology unit, and myself at IIIT Delhi. So we were looking at multiple myeloma, which is a type of blood cancer, and this blood cancer is actually a typical cancer where there is so we say that when it is a full blown multiple myeloma, we call it as a malignant stage, not healthy stage, malignant stage. But this cancer is typical in the sense that it also has a benign stage, where it's not symptomatically, the patient may not be exhibiting all symptoms of multiple myeloma, is actually having those genetic changes have started happening, and which can be characterized as a benign multiple myeloma stage. So this is a normal B cell. So I don't have, so you can see my mouse. So this is normal B cells. MGUS is, we call it as monoclonal gammopathy of undetermined significance, because it is the monoclonal protein because of which this multiple myeloma cancer happens, and this is a benign stage. Smoldering multiple myeloma is the second stage, and finally multiple myeloma, which is the full blown cancer stage. Now typically what would happen is that maybe a patient is walking in the clinic when the symptoms are more apparent, or is falling sick because of some reasons, and the clinician may also be able to diagnose that the person is suffering with perhaps multiple myeloma, needs to go for the blood test. An MGUS patient, because it is a benign, at this stage the cancer is benign, may not exhibit that kind of a thing, so those patients may not even come to the clinic, and you may not have data of those patients. That's the first challenge. So you will of course assume that there will be a lot of data of multiple myeloma, there will be less amount of data of benign stage, but what becomes is a problem which is from clinical aspect really important is that you wouldn't like those patients who have MGUS should progress to multiple myeloma, should progress to the malignant stage. So if you say that I want to do early disease diagnosis and want to cure there, then and there itself, so that means you should be able to actually detect at MGUS stage and then should be able to cure that. Only then you can say that you can actually cure even before it moves to malignant stage. But the challenges, the real world challenges would be that you won't have much data of because of MGUS, because the patient may not come at that stage to your clinic. And the second would be that if you really want to build a robust thing, you will say which patient has actually progressed to multiple myeloma, but maybe a patient gets diagnosed because it is benign, would go back and may not come back to the same hospital. So the hospital may lose that patient. Longitudinally, you may not have that data. Maybe the patient dies because of some other comorbidity, not because of multiple myeloma, and so on. So you will see those challenges also. So the key research questions that hospital gave us was, can we identify genomic events responsible for transformation from MGUS to MM and the progression responsible for progression in multiple myeloma? And the second was, can we build such a genomic assay in the clinical setting that can come out in the market so that it can now be easily diagnosed? So these were the two research questions. And what were the challenges? One, of course, I have told you. So the existence of majority of the genetic abnormalities known in multiple myeloma, they are actually seen at both MGUS and MM stage. So normally what you will do is you will look at the entire literature, see what genomic abnormalities are known at MGUS stage, known at MM. So you are finding that most of them are actually seen at both. So distinguishing MM from MGUS based on the genomic signatures becomes a challenge in that case, and defining those clinical genomic events responsible for progression is another big challenge. The second challenge, lack of paired sequencing data, means in the sense the same patient which actually progressed from MGUS to MM, that amount of data would also be less. And now the data is also less of MGUS compared to MM. So looking at all those challenges, when we received the problem, it was a bit difficult for us, and but we wanted to see whether we can do justice with that. Now hold on with me on the slides and see as I am explaining, and see what if you get such a problem as an MLDL engineer and you receive such challenges, what you should be doing, what we did, and see whether you are able to connect with me that perhaps you could also do something of that sort. So the first step that we'll do is, let's collate all possible data that I find in the public spaces as well as at my own hospital. So we looked at MGUS and MM, and we looked at all possible repositories. So there is Multiple Myeloma Research Foundation that actually houses a huge amount of multiple myeloma data of different modality, in the sense whole exome sequencing data, whole genome sequencing data, RNA sequencing data, laboratory data, clinical data, as well as the disease, the treatment that was imparted, and longitudinal data. Whole amount of data is there. Although the number of patients at that time when we took this data was around 1092 patients data. So look at the amount of data. So can it be set as representative of the entire population that can have multiple myeloma is another challenge, but still it was a huge amount of data compared to what we could collect. So 1092, but they were all multiple myeloma patients, American population, source was MMRF, and we could receive after, it is with the controlled access, after writing the project and fulfilling their requirements, we could receive the data. Then we looked at the European data sets, whether we can access some of the data from there. So fortunately, we could access now. See, in the above one MMRF, we didn't have any MGUS patient data, only multiple myeloma. Then from the European consortium or different data sets, we actually, this is European genomic collection of the data, we could get data of only 33 patients. Then we collected some data in-house, All India Institute of Medical Sciences, they get lots of patients throughout the year. So they also collected data. So they collected data of nearly 82 MM patients and 28 MGUS patients. Now see what is the amount of data, overall data, 1154 MM samples and only 61 MGUS samples. Look at the kind of imbalance that it has. 95% data is of MM population, only 5% data of MGUS. If I am building a simple classifier and I say, let me build a classifier that looks at the features of both and does the classification, maybe the key features that distinguish both the population are the key features that I should be looking at into the assay. If it gives me 95% accuracy, I would be happy, but that means my classifier doesn't have to do anything, just say healthy, everyone is MM, and will do the job. So in the nutshell, the message is, even a simple metric like accuracy may not be the right metric. A simple model may not be the right model. You need to look at what should be your metrics, how you should be processing data, so that it gives you the really the robust results. But the first and the bigger point was how should we process the data, because what we were processing was the whole genome sequencing data, which is actually and whole exome sequencing data, so of the coding region and the non and all data. So when you look at only the coding region data, that translates to proteins, right? So coding region data, that is actually only 1.5% of the whole genome. So we had both types of data, and we wanted to, and this is whole genome is billion long AGCT nucleotide pair sequencing data. How should you process the data where you are defining genes, you are looking at the chromosomes where the genes are sitting and the mutations are happening in the genes. That is the kind of data we were analyzing. So the first is how should I extract features out of that and then start processing the data. So what we did was we looked at what all possible ways these kinds of genomics data are analyzed by the genomic or the bioinformatics community, and what as an ML/DL engineer we can do on top of that. That was the idea. We should understand how they process it, and we should also know how we can add more information and how we can process it. So if you look at the first box here, we are saying that we have whole exome sequencing data. Largely it was whole exome sequencing that even Ames also collected. And so we have from Ames, MMRF and EGA, and we collated all these population data set. Now in the second box, which is on the right, this complete box where we are saying WES data pre-processing, this is actually how a traditional processing happens in the bioinformatics domain. So what happens is that you look at so there is a reference genome. So we have a reference genome which is of human reference genome. We do have that, and whatever sequencing data we collect of a patient, we align it with the reference genome, and then we find out at which nucleotide any mutation has happened. So this way you detect the mutations. So if it is a single nucleotide that has changed, we call it as a single nucleotide variation or SNV. So what we did first, and there are a number of tools, standard tools which are used now to do SNV detection. So we did SNV identification using multiple such tools, so that because every tool would have some nuance here and there, and we didn't want to lose out anything there. So that is why we used multiple SNV detectors and then collated information out of those, so that no SNV is lost out because one tool may not be detecting it. So that's the first step, that we used 4 different SNV detectors to do that. Then we used another tool that was doing some SNVs or some nucleotide variation or mutations might be leading to some malignancy or are known to be pathogenic in nature, that they can cause problem. Some are known to be benign. So we did SNV filtration based on another existing standard tool, that benigns were filtered out and pathogenic were taken in. Then we did the annotation, then the annotations of SNV happens, and then another standard algorithm was used that was also looking at those SNVs. They are actually, these nucleotide or mutations are happening in the genes, so they are part of the genes. So then it is ranking the genes, that these genes are heavily mutated in different subjects. So we looked at MGUS cohort, we looked at cohort, did the ranking of those genes where most of the mutations were happening, etc. So based on the statistical algorithm, we found 362 significantly altered, we say statistically significantly altered genes in MGUS and altered genes in and some genes were common in both the cohorts. So that was this is a standard workflow that any bioinformatician would follow, and will look at those genes and then will be analyzing only those genes, will perhaps stop there. Now from here, we started our work that let us now enhance this workflow so that at the end we get something. So we did the union of them, and now, so there are databases that look at gene-gene interaction, and so there are number of such databases. So we said that, so besides genes in human biology, we also talk about biological pathways. So there is a cell signaling pathway. So there are number of pathways these genes are part of. So what happens is that when a gene gets a mutation, because it is part of one particular pathway, so it actually starts doing alteration within that, because maybe the genes is, this gene, because of its alteration, it is actually producing more protein or less protein. We call it as upregulated or downregulated gene. And that way, because these proteins then are in turn triggering different events within the body, so they start altering those biological pathways. So, and that is why we look at gene-gene interaction, because in one pathway there would be multiple genes which are part of that pathway. So when one gene gets altered, it starts impacting other genes in the same biological pathways, and a whole lot of things start happening. So now we looked at this gene-gene interaction. It is kind of, for an engineer like us, we'll call it as a similarity matrix or an adjacency matrix, which is stored in a database and we can access that. So we said, okay, fine, we'll look at this gene-gene interaction, which is already part of these databases, which is irrespective of the disease. But just a gene-gene interaction, this is how it is there. Again, we took it from multiple, we call it as protein-protein interaction databases or PPI databases. We looked at the 9 best, whatever we found, 9 best PPI databases. We considered the interactions from there, and then we took the union of those interactions for the genes that were part of our cohort, the union of 362 and 617. There were nearly 824 genes. Out of those 824 genes, roughly 26 genes were isolated, and rest 798 were connected in one or the other databases. So we took the union of their connections, and that way we downloaded this union as the protein-protein interaction or gene-gene interaction. Now you can imagine how I would think, I have one gene-gene interaction there with me. I can actually extract some features. A natural strategy would be for me as to think is, how about using some graph convolutional neural network that can make use of this graph gene-gene interaction, which was the natural thought process that triggered in, and perhaps that can exploit this gene-gene interaction as well as do this part. So we looked at the union of these, and we looked at those which were interacting. I already said that around 26 genes were isolated. So we filtered them out, and we were left with 798, of which 351 were part of MGUS and 598 were part of cohort. Then we created a graph convolutional neural network. We extracted features for each gene now. At each gene level, we extracted features. What could be the features like? We looked at SNV, single nucleotide variation. That was the only thing that we had. But basically, they have different categories like synonymous, non-synonymous, which part of the region they are, missense mutation, etc., etc. A lot of different classifications are there. Moreover, when you annotate them, you annotate them with some scoring. So those features, then within a gene, how many synonymous mutations were there, how many non-synonymous, because now we are collecting these features at a gene level, not at an individual SNV level. So at each gene level, so we make a graph neural network, and in that graph, each gene was made one node. And at each gene, we constructed a feature vector that was a 28 cross 1 feature vector, which had total number of SNVs, total number of synonymous, total number of non-synonymous, then there is allele depth, variant frequency, allele frequency, lot of other features are there which are biological features. We collected them and we stacked them as a 28 cross 1 feature vector. So now I have a graph which is having 798 nodes. At each node there is a 28 cross 1 feature vector, and these nodes are connected with the known gene-gene interaction. That's how they are configured. And now we said that we actually had only 2 layers of GCN on this, and then we reduced the number of features at each node to 2, and then took them, did the collected all those features, and then we fed that to a single fully connected layer and did the classification of MM versus MGUS. So that was the simple architecture we set in, and then we did the classification of a fully connected, 2 layers of that. But in between, we also incorporated the idea of multi-head attention. So how we did that? We constructed adjacency. Now what we were doing was, we initialized the adjacency of this GCN with the PPI database, but then we said that we'll update as the GCN will get trained, we'll update this adjacency, and we are initializing 3 different adjacencies with the same thing, but then updating them separately, and they will be updated, and then a consensus matrix will be formed, and then the features will be propagated, and then the loss would be propagated backward and the network would be trained. So this is how we did that. So we said that we put multiple head attention with the consensus which is happening on these adjacency, and in the end it does the classification. Once classification is done, and of course we had cost sensitive classification in the sense that the cost of minority class was minority error is given higher weight as compared to majority class error, and that way we trained the network. But still we wanted to do the post hoc analysis as to whatever we are doing, is it making sense or not, but still a lot we did in that. So we did post hoc analysis of this model. We called it as bio-DGI model. We did Shapley analysis. We also did, because in the end it gave us the adjacency, updated adjacency, and on updated adjacency it is nothing but a graph. We could do the community detection, run community detection algorithm, or in the sense that subgraph analysis, and then again looked at those analyses. I will show you later. We also took some other features which are copy number variation, structural variants, etc., which actually come from WGS data alongside, and we incorporated all that information eventually to build a 295 gene panel, which is actually a good panel in the sense that kind of a assay that can come out in the market. But see how we did that. So once we have trained the model, graph neural convolutional neural network, and now we have sort of updated adjacency, but because we did 5-fold cross validation, so that means we had 5 classifiers and we had 5 such adjacency matrices. We ran Leiden algorithm, which is a community detection algorithm, and then and every time we ran the algorithm, we took the top 3 communities. How did we identify the top 3 communities, most connected communities, as well as having larger number of genes which are known as oncogenes. Oncogenes are cancer causing genes. Some are already known there. TSGs, which are called as tumor suppressor genes, which become active as soon as cancer starts kicking. Tumor suppressor gene will get active so as to suppress the cancer. Oncogene will get more activated. So if TSG is not able to suppress, then oncogene are actually triggering the disorder moving towards progression. So there are some like oncogenes, tumor suppressor genes, driver genes, actionable genes, which are known for cancers, and there are databases where such collection of genes are there. You can actually fetch that information that these genes are known to be driver genes of cancer, at least in one or some other cancer. These are known to be oncogenes, these are known to be tumor suppressor genes. So now when we are looking at communities and which communities are important, so natural for us is to connect with the domain and see which community has largest number of known OGs, TSGs, ODGs, etc. Perhaps that community is valuable. So immediately that means the domain should go in and should help me with that data whether I should consider it or not. So this way we fetched top 3 communities, then generated a new learned adjacency by merging all of them, again applied Leiden algorithm and to find the final gene communities. So this is one analysis, and once we found those gene communities, we showed to the oncopathologist or the oncologist who is a specialist that do you see some genes which are acting as hub genes here, do they really make sense as they are really important for us to validate or to look at that? And they could verify that some of the communities were actually known as that the genes which are part of that, they were known to actually alter those pathways, critical pathways which are characteristic of multiple myeloma. So that way, so they listed as 5 features or 5 types of pathways that these are really key. When we look at multiple myeloma, we know that they get altered, and those genes are were really part of those top communities when we looked at that. So that is first cut of sort of validation. Indeed, OGs, TSGs are coming into part of that. Indeed, the genes which are otherwise, other than those OGs, TSGs, they are also relevant as far as domain knowledge is concerned, and an oncologist is able to endorse that, looks like it is correct, it is not that wrong, right? So then in order to make the panel, we further moved ahead. Okay, fine, we are able to find some good things, but let us see how we incorporate all possible information. So one possible information was we only utilized so far single nucleotide variants which we incorporated, but then there are also copy number variations. So what happens is that maybe multiple copies of a bigger chunk on the chromosome, they also built up, and so you need to identify whether such copy number variations are happening. So that was extracted, that information was extracted from by using different tools, and for the multiple myeloma MMRF data, it was already that processed information was available. For AIMS data, we did that. Then structural variants, where one part actually moves from one gene to another. We say, for example, translocation. So a gene or a part of that actually moves from, for example, T414, from 4 number chromosome it moves to 14 number, and those chunks are also translocations is a key in multiple myeloma. So we incorporated that information, loss of function of genes, and we then made sort of a thing that what we looked at the box plot, that how many median number of SNVs for every gene in MM cohort, and number of having non-synonymous or synonymous, then put some thresholds that if greater than this median is observed in a gene, will catch that gene and say that it is really important for SNV. That way we collected from SNV, CNV, SV, LOF, and then took the union of those genes. We got a panel of about 282, and then we also looked at the literature. Some genes may not figure in. So for example, if a translocation has happened from one to another region, so maybe that gene is not there, or deletion has happened of a bigger chunk, I will not see that gene in the SNV. So 798 genes, maybe that gene will not be visible as part of those 798 genes because that chunk was already moved, so you can't find that single nucleotide variant because it got either deleted or moved somewhere else, right? So that is why some genes which are known as the key genes for multiple myeloma, we looked at the literature and we also incorporated them, and that's how the panel became of 295. Then we did the survival analysis check, that if we have included these 295 genes, so we also had for multiple myeloma patients overall survival time. So some patients died, some patients were still surviving, but from the baseline when the disease is diagnosed until the time that the patient died, or until the time when the measurement was taken whether he died or whether he survived. So we looked at that survival data, and then at gene level, if it was having SNV mutation, yes or no, and then put it inside for a patient, for different patients, and looked at the survival analysis. And that way we validated for individual profiles SNV, CNV, SV, LOF, and we also combined them, and because they were these were different data, so we used factor analysis for mixed data analysis sort of a thing, and then again performed the univariate analysis on the principal component found there. So in two ways we did the survival analysis, again to figure out whether the genes that we are finding are really making sense or not. The MLDL model that we made, we called it as BioDGI, which was using 9 different PPI databases, but at the same time we also implemented cost sensitive decision tree, the first one, then cost sensitive SVC, then cost sensitive random forest, XGBoost. These were the traditional, and you find that most of them, if I am looking at the balanced accuracy, the light green, the light blue one, you will find that most of them are really high. So we found that the numbers, whether I am building decision tree, whether I am building random forest, XGBoost, or my own method, my accuracy is almost nearly same. And for the top 3 model, for example, CatBoost and this BioDGI with the attention based, etc., my numbers are almost similar. But still, how do I convince myself which one is actually a better? So what we did was that we looked at the number of OGs, TSGs, etc. that are found in using different classifiers in top 250 or top 500 altered genes, and we found that if you look at the lower bottom, we found that, for example, CatBoost was the third or one of the best models that was sort of giving the similar results. But in the top 250 ranked genes, there was no OG TSG. Even in top 500, there was no known OG TSG, ODG, AG. But with our model that we built using 9 different PPI databases, has the highest number of known OG TSG as part of those top ranked genes. Among those top ranked genes is another sort of level of validation. Indeed, whatever model has been found is making sense, is extracting the key things and not just random things. Otherwise, based on numbers, someone would have said, if CatBoost is working, then what is the point of building such an attention based network? It is doing a good job. So that way we validated that. Then we also did genomic feature ranking of this BioDGI PPI9 from the 9 databases as post hoc analysis, and then got them, we ran Shapley analysis at gene level as well as genomic feature level, and we got those validations again done from the doctors that whether the top scored features, they really make sense when it comes to this domain. If they make sense, that means we are extracting the useful information, otherwise something is not correct. We also looked at pathway enrichment analysis, which is part of sort of bioinformatics analysis, but we now looked at pathways which were increasing in significance as we moved from MGUS to MM, and pathways which decreased in significance from MGUS to MM, and pathways which were significantly altered in MM and not in MGUS. Now it was an interesting finding this time when we analyzed all the pathways that were altered at MGUS stage and MM stage based on the significantly altered genes that we found using our BioDGI model. It was interesting to see that at MGUS stage they were random, like not the regular MM related pathways which we found altered. So for example, thyroid pathway was altered, diabetic pathway was altered. So maybe those subjects were actually suffering with those kinds of comorbidities, but not the key specific MM specific pathways we found altered at MGUS stage. But they were really key pathways which were top ranking pathways in the MM stage. Now this gave us a feeling that at the benign stage, perhaps the body was getting destabilized little bit. It was getting the homeostasis of the body is getting destabilized, but by the time it was in it was hugely, when it was full blown cancer type malignant, at that time it was totally towards So that means that at MGUS stage, perhaps it could be arrested, it could be brought back to the normal stage, it was still not full blown malignant stage and could be taken care of. So that was an interesting finding, that we found different types of pathways altered at different stages, and the gene biomarkers panel that we made, this analysis was done using that, and indeed it could show that those pathways which were specific, they were actually increasing in significance as we moved from to MGUS to So for example, immunity is altered and immune system pathway, and these pathways, the top ranked ones, were really key pathways in cancer and human T cell leukemia infection kind of pathway, MAPK signaling pathways. So the doctors could immediately recognize top scoring pathways, that indeed these are the ones which we find altered in multiple myeloma. So these are some of the gene communities I am showing. So for example, we are circling the newer genes that we found that are not part of OGTSD with the magenta, and some of the genes, for example UBC and BPTF, the doctors, the doctors could immediately connect to, that they are known to be important, but they are yet not known as a known driver gene. So we also came up with a new terminology. We said that network influencer genes, perhaps these are influencing the networks within the system, but they are not yet known as a cancer driver genes, and perhaps they are the ones that one should be looking at. So we recently filed one patent on this, and we are now looking at wet lab validation to do the validation of this. We are building the panel, and then we want to do the wet lab validation where we collect data of prospective subjects and see that. So we also did GO2R validation, where we looked at the genes that we found as significant. Are they reported in the literature, and how many studies they have been reported, or how many number of validated data sets they have already been reported. So for example, 27 genes were reported in 77 genes were reported in 4 different validated data sets. So this shows that most of the genes that we found, they were also reported in 1 or 2 different types of data sets which are there. We also looked at CNV type distribution across chromosomes, and we found that that was also matching with the literature that is there. So then we also looked at the translocations. These are the structural variants, and what are the associated genes with them. These are specifically translocations, or these genomic events. They are known to be characteristics of multiple myeloma. So for example, translocation from 11 to 14 chromosome, T11, 14, 414, etc. Deletion 17p13, KRAS mutation. KRAS is a gene type, NRAS, LTB. These MYC rearrangement, these are known to be the key of multiple myeloma, and these are the associated genes, and we wanted to see whether those genes are part of our panel or not, have they come out or not. We found that the genes were indeed part of, 1 or 2 other gene was part of our panel. So that meant, and they were also associated with genes in interaction with some other genes. So that showed that our panel was again relevant when we are considering these different types of mutations or biomarkers or known genomic events which are part of this disorder. So then we also looked at the previously reported multiple myeloma panels. So we found that, for example, most of the studies have come up with the small panel size. So nowadays people are coming with a larger panel size, and because cancer is known to be a heterogeneous disorder, it actually, from patient to patient there is a huge heterogeneity, and even within a patient over a period of time the mutation profile changes and it brings lot of heterogeneity. So that means that the panel should be wide enough, incorporating a lot of genes so that it should be detected early on. or should be detected without fail. So that is how our panel is bigger. So it is actually 295, there is a typo here. So we like our cohort size was also larger on which we built compared to the known studies in the literature. We used both WES, microarray, all types of data and detected multiple profiles. And with our panel, for example, the first panel came up with 47 genes. Out of these 47, 19 genes were reported in our panel as well. 26 of the second panel were part of our panel as well, and so on. So we could see many genes reported by these different panels were also part of our panel. So in conclusion, we designed a targeted sequencing gene panel by identifying key genomic biomarkers of designed a GCN based architecture, did extensive benchmarking, not only I would say quantitative but qualitative also, in terms of looking at TSGs, OGs, etc., in different ways, looking at survival analysis also. In different ways we did the validation of that, and we identified gene communities with the influential community members using the learned adjacency metrics, which we updated during the training of the GCN, and we also benchmarked the targeted panel with the existing gene panels. So this study demonstrates feasibility and strength of deep learning algorithms in analyzing large genomic data set, identifying such genomic events, establish key genomic events that can impact clinical outcomes in multiple myeloma. Thank you. I acknowledge. So I'm open to questions. It's a bit different application, but you should look at, as a data scientist, we should be willing to work in any domain and look at any exciting data.
Thanks for the session. I have a simple question. So when will you actually use a 295 gene panel? Because we're normally only looking at biomarkers with like 9 to 30 genes. So what would be the application for the gene panel of a size as this?
No, gene panel would be used to, so for every patient you won't do the WGS or WES sequencing analysis. Once you make a gene panel which is large enough, which can capture heterogeneity, in the sense that any patient comes, maybe in one patient, one gene is mutated that is not part of your gene panel if it is small, then you will say the patient is healthy, but actually the patient is suffering with multiple myeloma. So your gene panel should be large enough so that no patient goes undetected. That is the use of larger gene panel.
And what does the two-fold survival analysis mean?
So we did survival analysis in two ways. One was we did survival analysis only looking at SNV profile, single nucleotide variation, separately for single nucleotide, separately for genes having CNVs, SVs, etc. And we also did analysis again by combining all those profiles, and then we did factor analysis and looked at the principal component and then did survival analysis on that. Both ways we found that whatever biomarkers or the things that we reported, they were really relevant if that leads to positive results in the survival analysis. In the sense, the patients in which those mutations are present, their survival is less compared to those patients in which those mutations were not there, their survival was more. So that is the meaning of validating with the survival analysis. That is the key in the in this genomic clinical domain.
Okay, so I have one more question. So typically in differential gene expression, which is done for mRNA expression datasets, we are using adjusted p-values for the thresholds. So I noticed that you're using q-value during assembly filtering. So is that something very similar?
Something similar, But this was not microarray data. So we have not done differential gene expression data analysis, which is done in DEX, right? DEX is done in microarray data. This is whole exome sequencing data. So here the analysis is a bit different, but Q value is something related to the P value only, and that is a valid statistical test. Thank you. Any other question? Looks like I couldn't inspire more on this application. Not many questions. Okay. Thank you so much.